Antibodies, or immunoglobulins, can be subdivided into different classes according to their structure. There are 5 classes called IgG, IgE, IgD, IgA, and IgM, each having different functions. The class of an antibody is determined by its heavy chains, and each antibody can be produced either as a cell surface antigen receptor on B cells or as a secreted molecule. The secreted form of IgA is a dimer of the basic 4 chain structure, and IgM is a pentamer. Naive B cells which have not encountered their antigen initially express IgM as their surface receptor. The heavy chain of IgM is called a mu chain, and a B cell makes a mu chain by transcribing its recombined VDJ gene together with exons for the constant domains of the IgM heavy chain. These are called C genes. C genes for the other immunoglobulin classes lie downstream of those for the IgM. An individual B cell can switch the class of antibody it produces. This process occurs in the germinal centers of the lymph nodes, spleen, and other secondary lymphoid tissues. Class switching is accompanied by a second process, somatic mutation, which increases the overall affinity of the antibodies produced. A primed B cell enters a germinal center and undergoes rapid division. At the same time, a mechanism is activated which introduces mutations into the DNA around the recombined VDJ gene, so each B cell has a slightly different antibody. Occasionally, a mutation will generate antibody with a higher affinity for its antigen. Follicular dendritic cells in the germinal center hold stores of antigen on their surface. B cells compete with each other for the antigen, and only B cells whose mutations have produced higher affinity antibodies will compete effectively for the limiting amount of antigen. This is now processed and presented to T cells, which signal division and differentiation, but B cells with low affinity antibodies have no antigen to present to the T cells and die by apoptosis. The interaction with T cells also promotes immunoglobulin class switching. Look now at the heavy chain gene locus. The IgM C gene is preceded by a switching region which corresponds to switching regions preceding the C genes for all the other immunoglobulin classes except IgD. Here we see the event which occurs as a B cell switches from making IgM to IgG1. The C gene for IgG1 replaces the C gene for IgM, placing it next to the VDJ gene. Now the transcript from this encodes an IgG1 heavy chain, but with the same specificity as the IgM, since it uses the same VDJ gene which encodes the variable, antigen binding, part of the antibody. Exactly which switch will take place depends on the T cells and the cytokines they release, so the T cells determine what type of antibody response will be produced. B cells leaving the germinal center may become antibody-secreting plasma cells or memory cells. As a consequence of these processes, the affinity of the antibodies tends to increase during an immune response, and this is associated with a switch to production of IgG, IgA, and IgE. Secreted antibodies have many functions. The IgG antibody can act as an adapter. The 2 Fab arms of antibody have identical antigen binding sites. These form multiple non-covalent bonds with a specific antigen. The Fc portion of the antibody can link to Fc receptors on cells, including phagocytes and natural killer cells. This allows these cells to recognize their targets. For example, IgG can opsonize bacteria for phagocytosis by neutrophils. It can also activate the complement system, causing deposition of C3b. IgG is also transferred across the placenta from the mother to protect a child in the uterus and during its first few months of life. Meanwhile, other B cells may have switched to the production of IgE. This type of antibody binds to receptors on mast cells and basophils so that if they should contact antigen, they release inflammatory mediators. Other B cells, particularly in the Peyer's patches, may have switched to the production of IgA. IgA is produced as a dimer of the basic 4-chain immunoglobulin unit, linked by a joining or J chain. Cells of mucosal epithelia can bind IgA and transport it across to protect the mucosal surface. Some antigens, particularly polymeric carbohydrates, are not recognized by T cells and are called T-independent antigens. So the antibody response to these antigens shows neither increasing affinity nor class switching. The antibody against such antigens is predominantly secreted IgM, which is also the principal antibody in the earliest stages of an immune response to conventional T-dependent antigens. Because secreted IgM is a pentamer of the basic 4-chain structure, it is effective at cross-linking antigens and also fixes complement.
The main antigen-presenting cells are the B cell, the dendritic cell, and the macrophage. These cells can present antigen to CD4 positive cells. Additionally, all nucleated cells can present to CD8 positive T cells. All cells of the body express class I molecules encoded within the major histocompatibility complex, or MHC. Additionally, the B cell, the dendritic cell, and the macrophage express MHC class II molecules. These membrane molecules may contain degraded antigen peptides in a cleft on the top of the molecule, which they express to the T cells. The molecules regulate the way that the T cell reacts, since a T cell will only recognize foreign antigens when they are associated with the MHC molecules themselves. There are many varieties of class I and class II molecules, although in any one individual there may be up to 6 of each type. The MHC gene locus contains 3 class II genes, DP, DQ, and DR, each formed by 2 chains, beta and alpha. It also contains 3 class I genes, B, C, and A. Because each individual has 2 chromosomes carrying these genes, the maternal chromosome and the paternal chromosome, there are usually 6 different versions of the class I molecule and 6 different versions of the class II molecule, produced from the 3 different genes with the 2 chromosomes encoding them. Each of the class II molecules is formed from an alpha and a beta chain. The indent formed on the top of the molecule is where the antigenic peptide is held. Because the MHC is dependent on the genetic makeup of the individual, it is very unlikely that 2 unrelated people will have the same MHC molecules. Each of the class I genes produces a single alpha chain which folds itself up and interacts with another molecule called beta-2 microglobulin, which comes from a different gene locus. Since each individual has different MHC molecules, the performance of everyone's immune system is different. This affects susceptibility to diseases where the immune system is involved. Viruses are unable to multiply on their own. They depend on the body's own cells. To multiply, a virus must first enter the body. One of the ways it does this is through the mucous membranes of the gut or respiratory system. Some viruses spread in the blood around the body. They are not able to infect cells indiscriminately because they can only attach to cells expressing particular molecules on their surfaces. When a virus encounters such a cell, it binds using a complementary receptor. The virus is now able to penetrate the cell. The capsid, the part of the virus containing the foreign antigenic material, enters the cytoplasm. There it breaks down, releasing nucleic acid into the cell. It is important to remember that viruses can carry either RNA or DNA in single or double-stranded forms. Many viruses replicate in the cytoplasm where the biomolecules it codes for are synthesized. These include more viral nucleic acid, protein, and glycoproteins for the viral envelope if there is one, and viral surface receptors. As a side effect, it may also cause the cell to produce additional host antigens on the cell surface. Some viruses incorporate their DNA into the nuclei. The components of the virus assemble in the cytoplasm and at the cell membrane. Viral receptor proteins protrude through the plasma membrane, and within the cytoplasm, viral nucleic acids associate with the capsid proteins to form a new capsid. The plasma membrane buds off to enclose this new capsid, causing a new virion to be released into the body. Many virions are produced simultaneously in each cell, and the virus proliferates. Each released virion is capable of infecting another cell. Neighboring cells in the body are likely to be of the same type, and since the virus targets cells expressing specific host molecules on their surfaces, propagation of the infection is rapid. The immune system has several defense mechanisms against viral infection. Here, a virus binds to its target cell. As we saw, it fuses with the host cell and releases its genetic material into the cytoplasm. When the foreign genetic material enters the cell and starts to synthesize viral products, the cell is stimulated to make alpha and beta interferons. These interferons can signal to neighboring cells to directly inhibit viral metabolism. They do this by binding to interferon receptors, which signal the synthesis of antiviral proteins. The antiviral proteins can switch the cell into a virus-resistant state if it should become infected. Interferon gamma, produced by the activated T cells, also induces synthesis of MHC class I molecules, which make the cell more readily recognized by cytotoxic T cells. When a virus infects the activated neighboring cell, the antiviral proteins detect foreign genetic material, becoming activated, and block viral replication by breaking down the mRNA and stopping protein synthesis. These proteins put the cell into stasis, which limits further replication of the virus and spread of infection. This holding action gives the immune system time to mobilize the T cell response. One consequence of interferon signaling is an increased production of major histocompatibility molecules, particularly class I. This is synthesized within the endoplasmic reticulum, where it comes into contact with peptide fragments of all the proteins present in the cell. All of the proteins are sampled by binding to MHC class I, which carries it to the cell surface. This includes any viral peptides synthesized by the cell. T cells that have been educated within the thymus will recognize and bind to a receptor composed of MHC class I plus non-self protein peptide. It does this through its T cell receptor and CD8 molecules. Once attached, there may be a direct interaction between the CD95 ligand group of the T cell and a CD95 molecule on the target infected cell. An indirect signal may also be sent using tumor necrosis factor or lymphotoxin to a tumor necrosis factor receptor on the target cell. In either case, this triggers the release of antigens within the cell, which disrupt nuclear DNA and cause cell death by apoptosis. The cytotoxic T cell is now free to seek other similarly infected cells. Some viruses may attempt to evade immune responses by downregulating the production of MHC class I molecules. But a cell which lacks MHC class I becomes susceptible to killing by natural killer NK cells. The body's other defense mechanism is carried out by free immunoglobulin. Both infected cells and free virus present viral proteins on their surface. Antibodies can bind to viral proteins expressed on the surface of infected host cells. In this case, they signal to components of the complement system, which attack the cell membrane and damage the cell by membrane attack complexes. The same antibodies, on encountering a virion, can also bind to its antigens, making it unable to attach to and enter uninfected cells.